Hello. This first lecture, which is part of the series on MRI pulse sequences for neuroimaging research, is on the SpinEcho. In my talk about the basics of MRI contrast, I showed these two images, the left being a T1 weighted image and the right being a T2 weighted image. Now note, these are two different slices of the brain. And the source of that T2 contrast was that, as you recall, we have the magnetization vector aligned with the main magnetic field. And then we send in a radio wave along this axis here, and it creates a force and tips that magnetization vector perpendicular to the applied radio wave. And so it flips it around that axis. And if we apply the RF pulse just long enough to flip it into the transverse plane, we call that a 90-degree pulse. And then because of the interaction between the spins, they experience slightly different magnetic fields, and that causes these spins to dephase. And as a result, over time, the signal decreases. Now, actually, the spins experience slightly different magnetic fields for two reasons. First being the spin-spin interactions, and the second being more macroscopic non-uniformities in the magnetic field, or the B0 field. And as a result, what we actually have is what we call T2 star decay. That is, the relaxation rate, the 1 over T2 star, is the sum of the relaxation rate due to the T2 time constant and the relaxation rate T2 prime due to the more macroscopic magnetic field non-uniformities. So if we wanted to create an image just of T2, not of T2 star, how would we do that? The trick is by applying what we call a 180-degree pulse. As you recall, at this initial spin here, initial stage here, if we send in a radio frequency wave and have it on long enough, we can tip it into the transverse plane. We call that a 90-degree pulse. If we send in a radio frequency pulse now in the opposite direction, just same along that axis, what would happen? It would cause these spins, again, to rotate around that magnetic field. And if we apply it long enough that it flips them all the way to the other side, we call that a 180-degree pulse because it flips them 180 degrees. Let's take a closer look at what happens. If we have these three spins, the green one is sitting in a slightly higher magnetic field, and if we're processing a little bit faster, the red one a little bit lower, and the black even slower than that. You can see over time, because they're experiencing different fields, they process at slightly different frequencies, they get out of phase with one another, and the signal decreases. Now let's apply that 180-degree pulse. So that 180-degree pulse flips them to the other side. And now we turn off that 180-degree pulse, and the green one, it's still sitting in a slightly higher magnetic field, so it's going to be processing around the main magnetic field a little bit faster, and the red one a little bit slower. And so after a certain period of time, the signal starts to increase again, and the spins refocus and form what we call this echo here. Now, this is the situation if the spins were able to completely refocus, but we also have this T2 process going on, that is the spin-spin interactions. And because of that, the field experience in this half isn't exactly the same as the spins experience as a refocusing. And as a result, we actually have this signal decrease. So the first signal decrease here is what we call the T2 star. That's as the signal decays very quickly due to both the T2 and the T2 prime. There's more macroscopic magnetic field non-uniformities. And the center of this is just decreased by the factor of T2. Now, historically, the SpinEcho was actually one of the first ways that they could create an image or could create and collect the signals from an NMR or from magnetic resonance imaging. And the reason for that being that it's very difficult to create a very uniform magnetic field. In addition, it was hard for them to, with the early technology, to send in a radio frequency pulse and then switch and acquire the data and apply all the other gradients rapidly enough. So pretty much the signal was gone due to the T2 star decay before they could even acquire any meaningful signal. And so by applying this 180-degree pulse, they could lengthen the time between when they first send in the excitation and when they could start acquiring the data. Now, in terms of definitions, the time between the initial excitation and when you acquire this SpinEcho, we call that the echo time for the SpinEcho. So if we acquire our image here, this is what's called a SpinEcho image. And as you'll see in the lecture on K-space, if we acquire the center of K-space right here, the image would have just nice T2 weighting. And if we acquire the data here doing this T2 star, this is called what's called a gradient echo image and would have T2 star weighting if it has a long echo time. Now, if we don't want any T2 star weighting, we could just choose a very short echo time to try to minimize that contrast. Now we can also play repeated pulses, 180-degree pulses. So if we keep refocusing it, initially we see that the signal refocuses and then decays again. If we apply another 180-degree pulse, the signal refocuses and then would decay again and so on. And we could then obtain, with multiple echoes, a more accurate measure of this T2 decay. Now this sequence was first introduced by two papers, one from Carr and Purcell and the other from Myboon and Gill, and is sometimes referred to as the CPMG sequence. So hopefully I've given you some understanding of how we can get a T2 weighted spin echo image. This type of acquisition, that is a spin echo image, is actually being used more frequently for diffusion tensor imaging, as you'll see in